Professor Hyun-Duk Jang. She graduated Ewha Women University, and now she is the assistant professor of Seoul National University Hospital. The title of her topic is the CAP1 is binding partner of PCSK9 and is required for the degradation of LDLR by PCSK9. Professor Jang, please.
Thank you very much for a nice introduction, and I'm very honored to present my recent work in here. Today, I brought what's the mechanism of PCSK9-mediated LDL receptor degradations. LDL receptor enters cells, dissociates from LDL cholesterol in endosomes, and is recycled to the cell surface, whereas LDL cholesterol is directed to lysosomes for degradations. But if bound to PCSK9, LDL receptor is internalized and escorted to lysosomes for degradation through an unknown mechanism. PCSK9, I'm going to call it PC9 for inconvenience in this talk. Serine protease expressed by liver and intestine degrades hepatic LDL receptor for prevents LDL receptor recycling to the cell surface. Fewer LDL receptor results in higher serum LDL cholesterol concentrations. PCSK9 inhibitors prevents LDL receptor degradation, leading to reduction of LDL cholesterol levels. PCSK9 consists of three domains: prodomain, catalytic domain, CRD domain. Catalytic domain is known for LDL receptor binding. CRD domain, cis-rich domain, is known for resistin-like. What's the mechanism? Endocytosis and lysosomal degradation of LDL receptor after binding with PC9. 2014, Nobel's data presented very interesting story. PC9 CRD is crucial for lysosomal degradations. I already mentioned PC9 catalytic domain is needed for LDL receptor interaction, but CRD is needed for X molecule, but until now, and nobody knows what's the protein X. In 2007, PNAS paper, resistin homotrimer structure is very similar to CRD of PC9. And Samindi and Hughes Kim, 2014, and CAP1 is a receptor for human resistin and mediates inflammatory actions of human monocytes. I thought, is CAP1 a binding partner of PC9? And CAP1 is a molecular X? And I published it. CAP1 is a binding partner of PC9 and required further degrade of LDL receptor and this year. And editorial comments, CAP1, a tip of the CAP1 to cholesterol metabolism, and after well organized. And sequence homology CAP1 binds to resistin. PC9 has similar structural homology to resistin in its CRD. And direct interaction, IP was used to show a direct interaction between CAP1 and PC9 in mouse liver. And I also showed co-localized PC9, CAP1, and LDL receptor. And human genetic relevance, PC9 loss of function mutations. And we also performed the RNA silencing and generated knockout mice, in vivo model, and in vitro model for proving caveolin-mediated endocytosis. Here's comparing binding motif, resisting CAP1 and PC9 CAP1, and well interactions. With virtual modeling, 3D modeling of PC9 and CAP1 docking simulations, we also prove it. Here is direct binding between PC9 and CAP1. IP experiments and biopsy visualizing interaction between CAP1 and PC9 in living cells, far western blot of CAP1 and PC9, and finally we proved direct binding assay using SPR. Next, for which is with wild type PC9 and CAP1 variants, and wild-type CAP1 and PC9 variants. We proved the SH3 domain of CAP1 is very important, and CRD domain is needed for their interactions. CAP1 induces PC9-mediated LDL receptor degradation and increases the LDL cholesterol level. Treatment with PC9 nicely degraded LDL receptors, dose-dependent manner, and blocker with lysosome blocker and LDL receptors are protective. In 30 minutes, PC9 enters the cells in early endosomes. EA1 is the early endosome marker. And 240 minutes, 4 hours, which means 4 hours, and LAMP2 is the lysosome marker. And they co-localize LDL receptor, PC9, and lysosome. CAP1 is required for LDL receptor degradations. We performed knockdown experiments with CAP1 knockdown siRNA system, and treatment with PC9, LDL receptors are still protective. And LAMP2 is a lysosome marker in the case of CAP1 knockdown, and LAMP2 disappeared, and PC9 cannot enter the cells. And CAP1 knockout cells, we rescue experiment with the wild-type CAP1 and CAP1 variants. In AC deletion mutants, SH3 deletion mutants, LDL receptors are protective, which means it's not AC domain and SH3 domain is very important. Next, we generated intolerant-mediated CAP1 knockout mice, and they are embryonic lethal. And therefore, we performed experiments in hetero-knockout mice, and 2 months high-fat diet. No significant differences in LDL receptor mRNA level, but interestingly, LDL receptors are very protective. The protein level is very protective. CAP1 knockdown mice showed no big difference TG and HDL cholesterol, but LDL cholesterol is significantly decreased. And here is FPLC, VLDL and LDL cholesterol are decreased. We generated adenovirus of PC9 and inject IV injections and see the LDL receptor protein level and hepatic protein levels, very protective in hetero mouse. And with the PC9 virus, LDL receptors are degraded. And next, we see the human relevance. PC9 loss of function mutants cannot interact with CAP1. Here's a mutation of PC9 gene in human genetics. It's the GOF. Gain of function missense mutations in PC9 cause a rare form of autosomal dominant FH, hypercholesterolemia, and loss of function mutations in PC9 were associated with hypocholesterolemia. We generated A PC9 point mutants by site-directed mutagenesis at the CRD of PC9 among the well-known LOF and GOF variants. Red, GOF, and green, LOF. Interestingly, red indicated GOF and for LOF, and Q544 and H689 is not expressed here. It's among 4 LOF mutants, only S668, G670 could not interact with CAP1. And here is the SPR systems. PC9A514, which means GOF mutants, showed the stronger interaction relative to wild type, and LOF mutants showed weak interactions. What's the mechanism? Mechanism of internalization of LDL receptor, PC9, and CAP1 complex, covalent or clustering dependent. Until now, internalization of LDL receptor has been postulated as clustering-mediated endocytosis. Interestingly, in our left isolation experiments here is non-left part, here's left fractions, and see, this is PC9 proteins are relatively abundant in non-left portion. CAP1 is also. But after treatment, PC9 30 minutes, and PC9 is increased in left fraction, and CAP1 is also 60 minutes. Added receptors abundant in cytosols and come back in PC9 and CAP1 in the left portion, which are highly implied in caveolin-mediated endocytosis. So we're comparing between caveolin knockdown and clathrin knockdown. Here is caveolin knockdown cells. LDL receptors are protective, whereas clathrin knockdown cells, LDL receptors are well degraded. Next, we proved caveolin-1 binds to LDL receptor, CAP1, and PC9, which means quadruple complex in the liver of wild-type mice. IP assays in mouse liver. Here is multiplex immunofluorescence staining. Next, we comparing caveolin knockdown and clathrin knockdown. Come back with PC9 overexpression systems. CAP1 knockdown and caveolin knockdown, LDL receptors are protective, but clathrin knockdown, overexpression of PC9, LDL receptors are degraded. Here is immunostaining. Stain with LDL receptors with LAMP2 means lysosomes. LDL receptors are degraded after PC9 treatment, CAP1 knockdown and caveolin knockdown. LDL receptors are protective. In treatment with PC9, LAMP2 appeared, which means lysosome formation, and CAP1 knockdown and caveolin knockdown could not. But clathrin knockdown, LDL receptors are degraded and lysosomes is appeared. What's the role of clathrin-mediated vesicles? We stained with LAB1, as LAB1 means recycling endosome marker, with LDL receptors. And here, clathrin knockdown, and we couldn't barely see in the LAB11 and LDL receptors, and caveolin knockdown and PC9 is still C. So which means clathrin-mediated endocytosis is needed for recycling. So taken together, LDL receptor in the presence of PC9 is directed to caveolin-dependent endocytosis and degradation, while recycling depends on the clathrin-mediated endocytosis pathway. We observed the electron microscope caveolin formation after PC9 treatment. Here is normal Hep2, and after PC9 treatment, see this is caveolin vesicles and numerous caveolae. And red indicated clathrin vesicles. And here's CAP1 knockdown. As treatment with PC9, the caveolin and caveolae couldn't be detected. Only small amount of clathrin vesicles we can see. So next, our question is, this is specific event or general event? So here, EGF is known for clathrin-mediated endocytosis, and albumin is known for caveolin-mediated endocytosis. So CAP1 knockdown cells, and EGF-mediated endocytosis is still detected, but albumin-mediated endocytosis is defective in CAP1 knockdown systems. So which means maybe CAP1 is very important for caveolin-mediated endocytosis. It's not only for LDL receptors, but also for general events of caveolin-mediated endocytosis. So we published it in August 16th in European Heart Journal in a basic science paper. And here's the summary. LDL receptor bound to LDL cholesterol is recycling. Even interaction bound to PC9, also recycling. But only LDL receptor bound to PC9 and CAP1 and goes to the lipid left and goes to caveolin-mediated endocytosis and degradations. Here's take-home message. CAP1 is the new binding partner of PC9 and mediates caveolin-dependent endocytosis and lysosomal degradation of LDL receptor. Thank you very much for attention.
Thank you, Professor.